Hello, and welcome to our 7th lecture in cognitive neuroscience. As we continue looking at various forms of neuroanatomy, we're going to start looking at larger structures in the nervous system. In particular, we're going to start with an overview of the nervous system, or systems. Really, we have a nervous system that's divided into a bunch of other nervous systems. So we're going to start with an overview of nervous system structures, talk about the autonomic nervous system in some detail, and then finish out talking about the central nervous system. So let's start off with some overview of nervous system structures. This is a more fine-grained look at sort of the ways in which the nervous system is built. So we'll start by looking at some what are called microcircuits, then move up to neural networks, and then get to the divisions of the nervous system. So in microcircuits, these are generally localized circuits. So a single cortical neuron generally receives inputs from thousands of other neurons, 1,000 to 5,000 other neurons. So this would be a sort of local single microcircuit. So as we start to connect these into microcircuits, we get processes of specific kinds of information, and these accomplish a variety of different tasks. So for example, when we get to vision, we're going to talk about microcircuits that are contained within the retina itself. And these are important as they start to accomplish a variety of perceptual functions before neural signals have even left the brain. And so we start being able to identify edges, borders, textures, that sort of thing, by these microcircuits contained within the retina itself. So these are very localized microcircuits that provide early processing of information. These then move up to neural networks. So these are long-distance connections between brain regions that form neural networks. So we talk about connections between two cortical regions, and so we might refer to this as a cortico-cortical connection because it starts at a cortex and ends at a cortex. So when we start talking about cortical circuits, and we introduced some of these topics in the previous lecture on neurotransmitter systems, so things like the mesocortical pathway or the nigrostriatal pathway, describe where they begin and where they end. So the mesocortical pathway starts in what's called the mesocortex and then moves up to the association cortex. And the nigrostriatal pathway begins in the substantia nigra and travels to the striatum. And so it's important to understand how these neural networks are described, and we'll start looking at how they function. So for example, when we talk about attention later in the term, we'll be talking about attention networks that form a really important variety of tasks in the attentional systems. So just to give you an idea about what some of these might look like, so we have a sensory motor circuit, dorsal cognitive circuits, frontal parietal networks. So these cortical networks and connections start to form important connections across the brain. And in fact, this is an area of great interest and research in what's called the Human Connectome Project. We're starting to see that these connections between hemispheres and across the brain are an important part of understanding brain functions. And in fact, we've been able to see, for example, that poverty and stress have very significant negative effects on developing brain in individuals. And so we're starting to use these kind of modern techniques to look at this. So this is a white matter fiber architecture from the Connectome data set. And what we're seeing is connections from the brain stem to the cortex and some interhemispheric connections. And these are color-coded by the direction in which they're going, from left to right, anterior to posterior, ascending and descending. And we're going to talk about the technology involved in the Human Connectome Project called diffuse intensor imaging, which is a version of magnetic resonance imaging. And we'll talk more about that when we get to research methods here in the next couple of lectures. So that then gets us into thinking about how we divide up the nervous system. So we have a nervous system that's divided into a central nervous system and the peripheral nervous system. The central nervous system is the brain and the spinal cord. Everything else is the peripheral nervous system. So for example, your eyes and the optic nerve are actually considered part of the peripheral nervous system until they synapse at the lateral geniculate nucleus of the thalamus. We then have the autonomic nervous system and the somatic nervous system. The somatic nervous system is going to control movements of our skeletal muscles, but also receives sensory input from a variety of different sensors throughout the body, including things like the eyes, ears, etc. The autonomic nervous system is involved in controlling and regulating our body's internal environment. So sort of self-regulated internal organs and glands. It's then further divided into a sympathetic nervous system and a parasympathetic nervous system. The sympathetic nervous system is our arousing autonomic function. That is, it engages our fight or flight response. And so when we're engaged in high stress activities, the sympathetic nervous system is often involved, and we'll talk more about that here in a moment. The parasympathetic nervous system works in opposition to that, keeps us calm. These two are sort of usually in opposition to one another, and we'll talk a little bit more about that. So again, the central nervous system is the brain and spinal cord. If it's not the brain or the spinal cord, it's not the central nervous system. Peripheral nervous system is the somatic nervous system. This is essentially the body's nervous system. And the way the body interacts with the environment is the best way to think about the somatic nervous system. So muscles and sensory systems. The autonomic nervous system, again, regulates our body's internal environment, and it's divided into the sympathetic and the parasympathetic nervous system. In the next set of slides, we're going to talk about that in more detail. So here is the autonomic nervous system. Again, it regulates the body's internal environment. And the sympathetic and parasympathetic nervous systems generally operate antagonistically. And we're going to talk about antagonism in a variety of different contexts. So essentially, this is where we have two systems that are pushing kind of against each other. Essentially, one is exciting and one is inhibiting. And the sympathetic nervous system is essentially inhibiting the parasympathetic nervous system is inhibiting the sympathetic nervous system. Then when one takes over, sort of has a greater level of excitation, it dominates. The interesting thing, though, is when an antagonistic system, when one has been working for a while and then calms down, so to speak, the other system sort of takes over at a greater level than it might otherwise. So the parasympathetic branch is involved. Well, it's easier to talk, about the sympathetic nervous system because it's really the more active version of this. So if we have a stress response, let's do a sort of simple stress response. You almost get into a car accident. It's happened to all of us who drive. So what's going to happen is your pupils will dilate. It will stimulate your tear glands. It will inhibit salivation, increase the amount you're sweating. It will accelerate your heart rate, dilate your bronchi, decrease your digestive functions, secrete a bunch of adrenaline and cortisol, will decrease your digestive functions, and will inhibit bladder contraction, and will also inhibit blood flow to the genitals. So this kind of stress response is designed to basically take all of your resources and put them towards survival. So your pupils dilate so that you can gather more light and see more effectively. Increasing sweating, that occurs because you're wanting to give off as much body heat as possible because your muscles might be generating a lot more body heat. Your heart rate is going to accelerate and strengthen. Your bronchi are going to dilate so that you can exchange oxygen more efficiently. And it's going to take blood from your digestive functions and your genitals and other areas and put it towards your musculoskeletal system or your muscular system. The whole purpose of this process is to provide as much energy to your muscles as possible so that you can fight or flee. The parasympathetic branch is working in opposition to that. It will constrict your pupils, increase salivation, slow your heart rate, constrict your bronchi, increase digestive functions, stimulates bladder contraction, and you'll have blood flow back to your sexual organs. In fact, there are occasions where people have spontaneous erections after a sympathetic nervous system response because the parasympathetic nervous system takes over. Now, there are a couple things we want to keep in mind about the sympathetic and parasympathetic nervous systems. Norepinephrine is the primary neurotransmitter of the sympathetic nervous system. So this is our primary stress hormone. Excuse me. It will trigger the release of cortisol. So norepinephrine and cortisol are the two primary stress responses to are the primary responses to stress by the sympathetic nervous system. They have very different effects. So norepinephrine has been hypothesized to improve episodic memory function, whereas cortisol has actually been shown to diminish memory functioning. And we'll talk about cortisol and its deleterious effects on the brain throughout the term. Acetylcholine is the primary neurotransmitter of the parasympathetic nervous system, and so it's primarily involved in the parasympathetic nervous system. Remember, in previous lectures, I talked about acetylcholine esterase inhibitors, and so, for example, an irreversible acetylcholine esterase inhibitor will alter the functioning of the parasympathetic nervous system dramatically, and so that's something to keep in mind. So I want to make a quick note. This is going to come up in a number of areas throughout the semester. There are a number of drugs which increase norepinephrine, and we refer to these as being sympathomimetic. That is, they mimic a sympathetic nervous system response. And so drugs like amphetamines, cocaine, etc., can increase the amount of norepinephrine and will mimic a sympathetic nervous system response, which is why people will have dilated pupils, they'll be sweating, they will be unable to obtain an erection, they can get paranoid, heart rate can increase, etc. It's also the reason why these drugs can be toxic, because they're taking blood away from internal organs to move them towards the muscular system. So that gets us then to the central nervous system. We're going to talk about some sort of overall structures, talk about the organization of neurons, and then finish with talking about the blood supply in the brain. This is going to be important as we move through the semester and talk about individuals who have had ischemic strokes and lost functioning in parts of the brain. Let's start with this overall structures. The brain is surrounded by membranes known as the meninges. These include the dura, which is a very tough outer layering that is just underneath the skull. It's kind of weird. It's almost like a really tough paper bag. It looks just like a paper bag, literally. It's very strange. Some of you may have heard of the dura mater and not realized it. You might have heard of what's called a subdural hematoma. And a subdural hematoma is where there is a blood vessel that is leaking underneath the dura. That is its subdura. And so a subdural hematoma is a bruise where blood is collecting underneath the dura, and then that blood starts to pool and push against the brain. And so that's a medical emergency that can happen due to a head injury. So if you have any kind of head injury, you always need to go and have it looked at in the event that you have ruptured a blood vessel and have a subdural bleed. Underneath that are the arachnoid membranes and the pia mater. The arachnoid membranes can get inflamed and infected, and in fact that's what meningitis is when these meninges, these membranes get inflamed and infected, and that is also something you want to avoid. Fortunately, there are vaccines available to prevent meningitis, and any college student should have had their meningitis vaccine, because meningitis spreads through colleges like wildfire. So the brain also is floating in cerebral spinal fluid, and that flows through these ventricles, which are these areas within the brain. And so the ventricles and the fluid actually support the brain and support their structure, and are very important for maintaining the health of the brain. In fact, there's very recent evidence that has shown that when you are in very deep sleep, what we call stage 4 sleep, the cerebral spinal fluid is actually washing through the brain, basically trying to clean up. Essentially what they think is happening is that it increases the flow of cerebral spinal fluid. There are also issues where you can get problems with cerebral spinal fluid. It can build up in what's called hydrocephalus or hydrocephaly, and so oftentimes this cerebral aqueduct, you can see here kind of coming down towards the spinal cord, can get clogged, and so sometimes they will go in and put in a stent. This can happen in infants. It can also happen in older adults in what's called normal tension hydrocephaly, which can cause dementia-like symptoms. So the cerebral spinal fluid is very important and will come up in a variety of areas. That gets us into organizations of neurons throughout the brain. There are compact arrangements of cell bodies, which we call nuclei, and often have functional significance. So, for example, we'll talk a great deal about the lateral geniculate nucleus in the thalamus, which is the sort of first stop-off for visual processing after neurons leave the eye. We'll talk about the medial geniculate nucleus, which is involved in hearing. The cerebral cortex then is arranged in 6 layers, or laminae as they are often called. So there are layers of 6 neurons that have very important functional properties. So for example, the 4th layer is often the input layer into an area. We also talk about tracts. So groups of axons form tracts that connect regions of the brain. For example, we have the optic tract, which is the optic nerve also, which travels from the retina out the back of the eye, goes to the optic chiasm, where one tract will go to the same side of the brain, and the other tract will go to the opposite side of the brain. Axons that project from one hemisphere to the other form bundles, which we call commissures. So the anterior commissure and the corpus callosum are the 2 largest of these, where we start to see fibers traveling from one side of the brain to the other. And we'll talk in a few weeks about the cerebral hemispheres and what happens when either the anterior commissure or the corpus callosum are severed in what's called a split brain operation. So finally, the overall general organization we think about as myelinated, or white matter versus gray matter. And myelinated axons forming tracts are white matter, whereas gray matter is neurons arranged in layers. And so we often talk about white matter volume versus gray matter volume. So connectivity, basically it's a way of looking at connectivity versus overall processing. And we'll start to see some effects on gray matter versus white matter in a variety of circumstances. So that gets us to the blood supply in the brain. About 20% of our blood volume is pumped to the brain. The brain is very metabolically intense. That is, it uses a lot of glucose and oxygen rapidly, but it doesn't have any way to store those. And so any disruption in blood flow or disruption of oxygen can result in serious neuronal injury. So hypoxia, carbon monoxide poisoning, these are very serious issues that can cause significant brain damage. We'll talk about some specific cells that are susceptible, like the pyramidal cells of the hippocampus, as we move through the semester. So that disruption in blood flow or oxygen often can result from ischemic stroke as well, which we'll talk about here in a second. So blood flow of the brain is related to the metabolic demand of neurons. So neurons use a lot of glucose and they use a lot of oxygen. Remember, if we have to go back to the Krebs cycle uses glucose, and neurons use a lot of adenosine triphosphate, or ATP, particularly that sodium-potassium pump we talked about a couple lectures ago. So the brain's very metabolic, has a high metabolic demand. And this is one of the ways we measure brain activity, by watching blood flow in the brain through what's called regional cerebral blood flow. We can also track where the brain is using glucose using positron emission tomography. Finally, ischemic stroke is the primary cause of brain injury throughout the world, generally caused by hypertension and aging. So here is a general look at all of the major blood vessels we often talk about. Usually we're talking about blood supply to the brain, so arterial flow, less than we are talking about veins, because veins are, of course, where blood is being removed from the brain. So we're going to talk about getting blood to the brain. Just want to take a closer look at some of these. So here over the internal carotid artery, this is the main blood supply. Then we get cerebral branches. In fact, the middle cerebral artery is one we're going to talk about quite a bit, because this is an area where there is often stroke issues, and these can result in language disruptions. We'll talk more about those later on. We can look at the internal mid-sagittal view of these particular arteries. So we have this internal carotid artery, basilar artery. And so if you know where someone might have had a stroke, you can then look and see where they might have loss of functions by looking at where it is on this particular kind of map. So finally, this is a view from the bottom of the brain, and one of the things that gets talked about often is the Circle of Willis, and this is an area where we will revisit in a number of areas. So the blood flow to the brain is particularly important, and knowing where someone might have some sort of blockage is an important part of understanding how they might have lost some sort of function and what part of the brain might be damaged. All right, well, that is a quick introduction to the nervous systems. We will be talking about navigating our way through the brain in the next lectures.